lecture on contributions of scientists. In this lecture, we will be covering the period between 1935 and 1967. We will start with the contributions of Stanley. In 1935, Stanley actually proved that TMV is not a typical living organism. He crystallized this TMV, tobacco mosaic virus, and this is where you can see the viral particle inside the veins of the tobacco leaf. But actually, it is causing infection when inside, but outside it is a non-living entity. So that proves that virus is in between living and non-living organism. Next is the discovery of Thiller. Thiller produced a vaccine, the 17D vaccine against the yellow fever virus, which is the flavivirus. Flavivirus is dangerous because it is invading the host cell, utilizing the machinery of the host nucleic acid, and actually after replication it is utilizing the secretory components, that is the Golgi ER component, and it is released outside the cell. So he observed that this flavivirus is weak in monkey and they actually infer resistance. And on the basis of this particular observation, he discovered the famous yellow fever vaccine, and actually it saved a lot of lives. Next we come to the discovery of Biddle and Tatum, who showed in Neurospora crassa of fungi that the gene is controlling the activity of one enzyme, and this theory became famous as one gene, one enzyme theory. You can see when this particular fungus was irradiated with X-ray, they produced large number of mutants, and these mutants are actually controlling the production of the arginine from the precursor via ornithine and citrulline. So each time one of these mutants will be lacking the activity of one enzyme, and that they said was because of the mutation of one particular gene. So accordingly there is the wild strain which can produce naturally arginine, while the others they cannot produce arginine because of the dysfunction of one of the individual genes in this pathway. So this theory was called one gene, one enzyme theory. Of course, later on, Ingram proved that one gene cannot control the activity of entire enzyme. Rather, it is said that one particular gene is modified at a particular location, and that is point mutation, which is bringing about the modification of an oligopeptide. As you can see in hemoglobin, that normal one gene is actually related to one oligopeptide. That is, in case of the beta chain, the 6th position glutamic acid is replaced by valine, and that actually modified the hemoglobin structure, causing the normal sickle cell anemia. So thereby one gene, one enzyme theory was replaced by one gene, one polypeptide theory. Now there is another important discovery which is by George Hurst, that they said that virus, when they invade the human blood, they actually bring about agglutination. And this agglutination is brought about because of the fact that normally the RBC, they have their outer membrane sialic acid, which will be actually reacting with the viral particle, and that viral particle will agglutinate the RBC. So by seeing under microscope this agglutinated RBC, they could easily make out that there is some viral infection in the blood. Now this is a very interesting discovery in Nigeria carried out by Albert and others. They actually showed that if that particular antibody is tagged with some fluorescent dye, then this tagged antibody is binding with the bacteria Neisseria, and thereby you can actually locate the bacteria, and you can say this particular discovery opened a new vista, which is fluorescent microscopy, adding fluorescence to the bacterial cell. Now this is a typical structure of a T2 phage, that is the bacteriophage T2. This was denoted by Anderson and Luria, and this T2 structure typically showed, you can see it is having a binal structure. This binal structure having a geometrical head and a particular helical tail. So this structure was denoted by Anderson and Luria in 1942. Followed by the discovery of Hunget in 1943, he showed that the lumen of the cow contain some typical bacteria which can degrade cellulose. So cellulose splitting action is carried out by the cellulose splitting bacteria, which actually help the cow to digest cellulose, which we cannot because we don't have that microflora in our stomach. Now we come to a very important compound. You know, Waxman and his colleague in 1944, discovered a compound called streptomycin from a particular actinomycetes called Streptomyces griseus. This particular antibiotic is actually blocking the protein synthesis, blocking the 30S ribosomal subunit of the bacteria, and thereby it was the only drug at that time efficient against the dreadful disease of tuberculosis. So the discovery of streptomycin actually prevent a lot of people from dying while they suffer from tuberculosis. Of course, later on bacteria developed resistance against this particular antibiotic, but this is a landmark, and obviously these discoveries, they are awarded with the Nobel Prize. Now we come to the next important discovery, which is the discovery by Delbruck, Hershey and Luria. This is the discovery of the common cold virus. You can see this is a typical geometrical shape of the common flu virus, and there are some subunits present in the outer capsid, and these subunits actually they may be hemagglutinin, neuraminidase, but at that time it was not known. So they said that because of the modification of the protein subunit in this particular viral particle, these viruses are dynamic in nature. You cannot produce any vaccine, and even now vaccine against flu is very difficult because these viruses keep on changing their topology. Now we come to the next important discovery, which is by MacLeod and others. They actually showed that when a particular vaccine is being raised, it is not necessary to have the entire cell. You can have the subunit vaccine. Like in case of Neisseria, they produce the capsular polysaccharide. This is the capsular polysaccharide subunit which is used as a vaccine. So it is obviously the vaccine produced against Neisseria is with the help of the capsular polysaccharide, which is the sialic acid residue of the bacterial outer wall. So that subunit vaccine became famous in case of Neisseria. Now we come to the next important discovery, which is the discovery of sexual reproduction by Lederberg and Tatum. They showed that the two bacterial cells, the donor and the recipient, come close to each other. They develop a breach, which is the conjugation breach, and then this particular circular DNA, which is actually replicating, and after replication this circular DNA will enter the recipient cell. This phenomenon was called conjugation, and by this method they said that bacteria can also reproduce sexually, and thereby when this particular plasmid is reaching the recipient, they are modifying, the characters are modified. If this plasmid, they sometimes call it the fertility factor, if it is F plasmid, then obviously originally it was F minus and this was F plus. So by receiving this particular F plus factor, they became F plus again. So this discovery of conjugation indicated that bacteria can also reproduce sexually.
So I have a question. Sir, how do plasmids help in conjugation?
You see, bacteria first of all do not reproduce sexually in the proper way, because this particular plasmid is outside the central nucleoid. So naturally they can easily replicate and they can easily be distributed to the recipient. So this is how this particular plasmid, by replicating and entering the new recipient cell, will help in conjugation or will help in sexual reproduction. Got it? The next important discovery is in 1947, where Brown and his colleagues discovered that this particular bacteria, that is Agrobacterium tumefaciens, they produce the TI plasmid, which is entering the plant cell, which is causing the crown gall tumor of potato. It is received by the potato cell, and there it enters into the DNA of the potato, modifying the rate of division, and this is actually inducing this tumor like outgrowth in the potato plant or in plants of Solanaceae. So this particular disease, you can call it cancer of plant induced by bacteria. Poliovirus, which is a deadly virus, it could not be cultured in the lab. So Enders and Weller and Robbins, they discovered a new method of culturing this particular virus, and this was by utilizing the human embryonic cell, and thereby they produced the viral culture, in vitro culture in the lab, and it could be easy to do research against this particular important disease which is causing paralysis in human, poliovirus. Now, viruses behave in two different ways. One is they cause the lysis of the host cell, and the other one is they cause lysogeny of the host cell. In the first case, you can see that the virus, which is the bacteriophage here, is attacking the bacteria, causing the complete destruction or lysis of the bacterial cell. This was called the lytic cycle. And the other one, where the viral DNA or that viral nucleic acid enters into the host cell and it is getting into the host DNA, and thereby it is replicating along with the host DNA. This type of cycle, where the viral DNA is integrating into the host DNA, is called lysogenic cycle. So this lytic and lysogenic cycle in virus was denoted by Loff and Siminovich in the year 1950. Bacteria is growing in nutrient, agar or in a medium, and it was two important scientists, that is Joshua Lederberg and Escher Lederberg. They developed a new technique which is called replica plating. They took a bacterial plate, and this bacterial plate was placed on a typical velvet paper, and this velvet paper was later on transferred to a new petri plate containing the medium, and thereby the number of colonies or the pattern of the colonies in the original plate was replicated. So two things happened by this way. One is rapid replication of the same bacteria. Number two is this particular bacteria could develop resistance against a particular antibiotic. So it was also shown if the later plates had antibiotic, some of the bacteria survived. So thereby they concluded that bacteria also can have the ability to mutate spontaneously. That came out of this experiment of Lederberg. And at the same time, Joshua Lederberg continued his work in bacteria, and he was the person who named the circular extranuclear DNA as plasmid. So the naming of plasmid was given by Lederberg, and subsequently he continued this work where he actually showed how that particular bacteriophage is replicating on a bacterial cell. The mechanism he studied very carefully, and he actually saw that these different stages of viral infection on the bacterial cell utilize the bacterial DNA, and they produce the viral DNA from the bacterial DNA, and subsequently these new phage particles will enter into another bacteria. Transduction is the method by which the bacterial DNA is moving from one bacteria to other via the phage particle. So this was discovered by Lederberg and Zinder. At that time, bacteriophage infection of bacteria was well known, but using this simple replication of bacteriophage, Hershey and Chase actually discovered that the DNA is the genetic material. They labeled the DNA, and at the same time they labeled the protein of the capsid of the bacteriophage, and they showed that in case of the labeling of the DNA, the radioactivity was coming up in the next generation of the viral particle, but there was no radioactivity detected when the labeling of the sulfur in the protein of the capsid was done. So thereby it was said that the infection and replication of the phage particle is brought about by the nucleic acid, and in other words it was said that DNA is the genetic material. Now penicillin discovery already had taken place, but how it actually prevent the bacterial growth was demonstrated by Park and Strominger. They showed that actually penicillin blocks this polypeptide linkage or tetrapeptide linkage in the cell wall production of the gram-positive bacteria. So mechanism of penicillin action was determined, as a result of which they also could understand why this particular bacteria can develop resistance against penicillin, obviously by producing penicillinase. Polio vaccine developed by Salk. Salk vaccine was actually prepared from inactivated poliovirus, and they utilized three different wild virulent strain, namely the Mahoney, the MEF and the Sauket. By utilizing this particular vaccine, they actually prevented the dreadful polio disease. So Salk vaccine was very important as the original vaccine against polio, but it was injectable. Of course, later on the oral polio vaccine was discovered. This is the landmark, the discovery by Watson, Crick and Wilkins. They utilized X-ray crystallography to show that the DNA double helix is formed of two different components, where it looks like just a spiral staircase, and this right-handed plectonomic coil has phosphoric acid and pentose sugar in the strand and the nitrogen bases as the steps. So this particular DNA structure or DNA double helix model was denoted by Watson and Crick in the year 1953, and subsequently they got the Nobel Prize in the 60s. The discovery of Watson and Crick, DNA, actually proved that there are specific components of DNA, and that actually was extended by Benzer. Benzer actually showed that specific areas of DNA having functional quality is called gene. So he accordingly showed that some gene fragments are responsible for mutation, some gene fragments are responsible for recombination, some gene fragments may be responsible for replication. Accordingly, different terminologies were given, which were called muton, recon, complon, replicon, etc. So this functional integrity of a gene is present within the functional DNA segment commonly called cistron. Coming to the next important discovery, that is the discovery of interferon by Isaacs and Linderman, which is a glycoprotein and having typical folded helical structure. This interferon is produced in a particular animal host under the infection of virus, and it was shown that interferon can actually inhibit the viral particle. So interferon producing gene, if it can be transferred from one animal to other, can actually be a definite answer to viral infection. And how do they actually inhibit viral infection? It is via the degradation of mRNA by activating enzyme ribonuclease. And at the same time, it can activate specific protein kinase, and that actually will bring about the phosphorylation of the initiation factor and inhibit protein synthesis. So this particular discovery is definitely indicating that animal can produce protein which can inhibit protein synthesis. Coming to the next important discovery by Gajdusek, which is a folded protein called prion. And this prion protein can actually induce infection, and they cause the deadly disease of mad cow disease or the scrappy disease or the kuru disease. But they were initially believed to be virus, but later on it was shown that they are not the virus, but they are the infectious protein, and thereby they are called the smallest infectious particle in the living world.
Sir, I have a question. Why are prions called slow viruses?
Actually, viruses can invade and can induce infection faster, but prions, because they only have the protein, they take time to develop within the host cell and to control over the host cell metabolism. So naturally this incubation time increases, and thereby the action becomes slow. So they are rightly termed as slow viruses. Now we come to the next important discovery by Burkholder and others, and which is actually the discovery of this fluorescein isothiocyanate. This FITC is actually a fluorescent compound which is tagging the antibody, and thereby this antibody attached to the bacterial cell will help the bacterial cell to fluoresce. So this technique of using FITC tagged bacteria is also helping us to see the bacterial cell by fluorescent microscopy. Now DNA replication, the method of DNA replication was already there, but it was Messelson and Stahl in 1958 who indicated that DNA replication is semi-conservative in nature. They labeled the nitrogen N15 in the DNA strand, and this radioactive labeling was followed by normal replication. So when the DNA was replicating at each generation, it was observed that the radioactivity is reduced by 50%. So it was said that one of the strand is conserved and one of the strand is produced after that, and thereby you can see that radioactivity is reduced by 50% after every generation, and that ultimately indicated that DNA replication is semi-conservative in nature. Now we come to the structure of E. coli plasmid, and this E. coli plasmid structure was denoted by Sawada. You can see it is a typical circular DNA molecule having specific origin of replication, and there are some important areas of bacterial resistance. The bacterial resistant genes are very important, and at the same time there are other genes, the insertion sites are there, which will allow this particular plasmid to act as vectors to transmit DNA fragments from one host cell to other. So this is a very important discovery which was done by Sawada.
So another question. So what is the utility of antibiotic resistant gene within the plasmid?
Yeah, you see, this antibiotic resistant genes, when a particular plasmid is used in say transgenesis, that is transfer of gene, this antibiotic resistance will also go. So naturally it will act as a marker gene. Thereby we can say we have successfully transferred a gene from one host to the other. Now the production of ATP is the next important discovery by Peter Mitchell, who is also a Nobel laureate. In 1959 he indicated that the production of ATP is via the ATP synthetase complex. It is a part of the electron transport system, and this electron transport system is having specific areas like NAD, FAD, coenzyme Q, cytochrome complex, etc. But as this electron is moving as per their redox potential, they open the proton gate, proton channel, and this proton will be accumulating at the base of the ATPase enzyme, and as they go in, they will activate the ATPase enzyme, and thereby ATP is produced. And this is how ATP production in living membrane was explained. This particular enzyme complex, what you can see, was denoted by Kornberg, and it is called the DNA polymerase enzyme. And it was shown that this particular enzyme has stator, motor and wrench subunit. This is helping in DNA polymerization in bacteria, and at the same time it is helping in the removal of the particular RNA primer after DNA replication is complete. So the discovery of Kornberg definitely indicate that DNA polymerase is a very important enzyme in DNA replication, and its molecular structure was also determined later on. This model which you can see here was denoted by Jacob and Monod in 1960. This model is called the Openin model or Openin concept, and it was shown that there in a particular bacteria E. coli there are structural genes which ultimately help in the utilization of lactose, but these structural genes are controlled by different promoter and operator and regulated genes. So lactose here initially will not be produced. By adding lactose, the repressor protein is blocked and the system is switched on. So in this way the structural hierarchy of genes within a bacteria was explained in the form of lactose operon, and further development occurred in the subsequent years. Spiegelman and Hall in the year 1961 developed this DNA-RNA hybridization between the nucleic acid of bacteria and virus. So this is definitely a very important tool towards molecular modeling of DNA and RNA, and it also proved that what will be the host and what will be the pathogen. Sabine, I already told you, actually developed the oral polio vaccine, and this is a very important discovery because now also we have this particular vaccine given to children to prevent the dreadful poliovirus. The discovery of genetic code dictionary is very important, and this was done by Nirenberg, Mathai and also Khorana. They actually showed that which particular genetic code is coding for which amino acid, and accordingly they separated all the 64 different codes, and they actually pointed out which amino acid is being coded by them, which included the initiating codon, the stop codon, etc. The binding of the F2 phage with E. coli was denoted by Nathans and Zinder. They showed that this particular virus is entering the bacterial cell by binding with the pili, and thereby it was ultimately shown that the cell-free DNA can be utilized by the viral RNA to produce the specific viral coat protein, and thereby the viral replication can continue.
So another question, can you tell us about the significance of F2 RNA?
The F2 RNA will invade the bacterial cell, utilize its DNA to produce the protein. So in one word, it is the main infectious tool which will help the virus to replicate without the entire viral particle carrying out the lytic cycle. Now this is Bloomberg, who actually denoted the famous Australian antigen, hepatitis B antigen, causing the hepatitis B disease. And this is infected liver cells having the viral particles within, and this is how this particular disease is propagating from one host to the other via blood. So this was denoted by Bloomberg. In the year 1964, a famous discovery was done by Holliday, which actually explained how this particular chromosome are getting exchanged during crossing over, and this recombination model, you can see how they are getting recombined at the time of crossing over, is explaining two things. One is the change in chromosome fragments helping in evolution, and at the same time it is also explaining how the gametes are different from each other. But the model is called Holiday model after the name of the discoverer. A typical disk diffusion test by which it was indicated that which particular bacteria is actually being stopped by the antibiotic, and this was denoted by Kirby and Bauer, and still now we follow this disk diffusion test as Kirby-Bauer test. Now there is another important discovery, we call it Ames test, as because it was discovered by Bruce Ames in 1966, which explained that bacteria has the normal mutability and some mutation can actually change the particular bacterial strain from one strain to other, and thereby this particular modification will indicate the pathogenicity of the bacteria, and this was called the Ames test. This is a typical potato tuber, and this is infected with potato spindle tuber virus. This potato spindle tuber virus was denoted by DNR in 1967, and this particular PSTV is causing the disease, and it also indicates that the infection is not carried out by the entire virus particle, it is carried out by the infectious RNA. Coming to the next important discovery by Thomas Brock, which is the discovery of this bacterial strain called Thermus aquaticus from the Yellowstone National Park. You can see here the yellow scum of bacteria, which is producing an enzyme called Taq polymerase, and this enzyme will be utilized in PCR, where this particular enzyme is stable even at a temperature of 100 degrees centigrade. And the last major discovery, which will be by Arbour, which is the methylation and demethylation of the DNA. You can see the methylation of the DNA normally occurs at the cytosine residue, and this methylation is commonly protecting the DNA molecule. We call it capping, and normally it is very important during transcription when the DNA is producing the mRNA and this mRNA is going from the nucleus to the cytoplasm. So methylation and demethylation are very important steps in the processing of the RNA, or it occurs in the cytosine residue. So we will conclude today's deliberation. We have understood that what are the important contributions of different scientists between the period of 1935 and 1967. We have started with the discovery of the TMV, and we have ended with the methylation of the DNA, and maybe in the future we will continue this contribution by other scientists.